Hello, my name is Andrew Calle, and this presentation is to fulfill the requirement for my neuroscience honors application. I'm a senior at Johns Hopkins, and today I'll be presenting on my project, which was assisting in a publication done by my lab, the Kornberg lab at Johns Hopkins. And the publication's title is The Protein Kinase C Modulator Bryostatin 1 Therapeutically Targets Microglia to Attenuate Neuroinflammation and Promote Remyelination. And that's a mouthful, but I'm proud to say that we recently published this paper in Science Translational Medicine this past January, and my lab mentor, Dr. Payam Garabani, is the first author on this paper. And once again, this presentation is to highlight my contributions and what I've learned and my project, which was only a small part of this whole project. But yeah. I would like to give a quick overview of my talking points. So I'll be starting off with the background and objectives of just the basic knowledge and setting the stage for my project. And then I'll go into the methods and results, but they'll be highlighting specifically my project and my contributions. Then I'll talk about some conclusions, and then I'll get into my reflection, my personal reflection, and talk about future directions for this project. So let's start off with an introduction to multiple sclerosis. So multiple sclerosis, or MS, is an inflammatory demyelinating disease that affects the central nervous system, or the CNS, and it most often begins with a relapsing course called relapsing remitting MS, or RRMS. And RRMS is defined by discrete episodes of focal CNS inflammation, and they're followed by an unpredictable recovery that's often incomplete. The inflammatory attacks that are seen in RRMS are caused by the peripheral adaptive immune system. And in these attacks, dense lymphocytic infiltrates cross the blood-brain barrier, or the BBB, and produce plaques. And then these plaques lead to demyelination. And many people with RRMS eventually progress to secondary progressive MS. This causes a worsening of disability, for example, fatigue and vision problems, and the loss of those discrete relapses. Current MS therapies mainly target peripheral adaptive immunity, which prevents relapses and new plaque formation. However, no approved therapies promote recovery via remyelination or slow the worsening of disability that occurs in the progressive forms of MS, at least not yet. This leads me to my next point, which is the role of microglia and macrophages, and how they set the stage for the research that I'm going to be talking about. So the progressive forms of MS follow a pathology that is mostly independent of peripheral inflammation, unlike in RRMS. These forms are characterized instead by chronic activation of CNS innate immune cells, and these cells being microglia and macrophages. These cells play dual roles in CNS injury and repair. And in addition to widespread activation of these cells throughout the CNS, many chronic MS plaques contain a rim of activated microglia and macrophages. And these plaques, or this rim, is associated with lesion expansion, continuous demyelination, and the progression of disability. My lab is translational medicine, so we don't do clinical research. So a question that quickly arises is, how do animal models display this, the models that we work in? And so in animal models of inflammatory demyelination, microglia and CNS-associated macrophages contribute to injury through various maladaptive or bad functions. Some examples of this are antigen presentation, inflammatory cytokine production, and the induction of toxic astrocytes. On the other hand, microglia and macrophages also play an important role in remyelination and repair by phagocytically clearing myelin debris generated from demyelination. They also contribute to the resolution of inflammation by secreting trophic factors that support neuroaxonal health. So basically to say that in animals, in mice, microglia and macrophages also play a dual role. And so a major goal of current multiple sclerosis drug discovery is the identification of treatments that therapeutically modulate microglia and CNS-associated macrophages to inhibit maladaptive functions like the ones I mentioned earlier, while also promoting regenerative phenotypes. And given this dual functionality, our lab seeks to explore potential therapies that could shift microglia and macrophages away from those harmful inflammatory roles and towards their beneficial regenerative functions. We identified bryostatin-1, a protein kinase C modulator, as a promising candidate. With all of that being said, we can start to talk about bryostatin-1, the topic of the paper and the drug in question. So bryostatin-1, or bryo-1, is a naturally occurring compound known to modulate protein kinase C, PKC, and PKC plays important roles in various biological processes, including cell proliferation, survival, and inflammation. My lab has previously shown that bryo-1 attenuates neuroinflammation and neurologic disability in the experimental autoimmune encephalomyelitis mouse model, or EAE, which is commonly used as an experimental mouse model for multiple sclerosis. Bryostatin-1 also has been shown to have a favorable safety profile in human trials of cancer, HIV, and Alzheimer's disease. It's a very promising candidate in all types of pathological research. We just happen to be looking into it for multiple sclerosis, but it's a very hot topic right now. And so in this project, we wondered whether bryo-1, which has been shown to cross the blood-brain barrier, an important requisite for a drug is that it can cross the blood-brain barrier. So the fact that bryo can do this is great. But we wondered whether bryo-1 could produce therapeutic effects on microglia and CNS-associated macrophages. This is validated by the fact that PKC signaling has already been implicated in microglia function. And so now, having said all that, I can speak about how I particularly investigated the impact of PKC modulation and how we found that it had downstream consequences on the modulation of myelin repair. With all of the background set in place, I can now talk about my methods, or the methods that I learned in the lab and were able to apply to this project. And basically, these are my contributions to the paper. So in terms of methods, my primary contributions involved sectioning mouse spinal cords into stainable segments using a cryostat, performing immunohistochemical staining, or IHC, imaging these IHC stained samples, and subsequently conducting quantitative image analysis using QPath. This is an image analysis software similar to Fiji or ImageJ. Immunohistochemistry is a crucial technique in neuroscience and many biological fields that allows visualization of specific proteins within tissues. In IHC, primary antibodies bind specifically to target proteins, and secondary antibodies tagged with fluorescent markers allow visualization under a microscope. For this study, we specifically stained for several key markers, but just to give a few examples, we have MBP, which is a marker of myelin integrity, CC1, a marker of oligodendrocyte maturity, IBA1, a general microglia and macrophage marker, and I won't name them all, but I'll get more into detail about those on the next slide. And these sort of markers enable us to visually and quantitatively assess the therapeutic effects of bryostatin-1 on neuroinflammation and remyelination. So now we can get into the specific results that were obtained from the methods that I specified. I'll start off by saying that although many of the microglial and macrophage signaling pathways activated by BRIO1 are known to support generation and myelin repair, the actual therapeutic outcomes can vary depending on context. So these experiments were focused specifically on whether the modulation of microglia and CNS-associated macrophages translated into enhanced remyelination in vivo. I was not directly involved in the in vitro experiments. I only know about the in vivo contributions because they involved immunohistochemistry, sectionaling, and data analysis on QPath, which all of those were involved in these figures A and B. The figures A involving immunohistochemistry to stain them, and then imaging to capture those images, and then B being the results of image analysis on QPath. So for these figures and for the next figures that I'm going to show after this, we used the LPC-induced focal demyelination model with BRIO1, so not the EAE model. These mice were given a focal lesion by LPC injection and then were given BRIO or vehicle treatment beginning on 2 days post-injury. So figures A and B demonstrate the systemic treatment with BRIO1 significantly increasing the number of oligodendrocyte lineage cells and mature oligodendrocytes within spinal cord lesions at both 9 and 15 days post-lesion. So in figure 1A, the oligodendrocyte lineage cells are visualized in green, while the mature oligodendrocytes are visualized in red. And as you can see in both groups, bryostatin 1 has far more positives. And so why does this matter? Well, this increased differentiation of the oligodendrocytes is crucial therapeutically since these cells produce myelin, which is obviously the protective sheath, which is important for efficient nerve signaling. And in conditions like multiple sclerosis, as I said earlier, these oligodendrocytes are damaged by inflammation. So promoting the maturation of these cells from their precursors helps facilitate remyelination and the restoration of nerve function. And our data shown here demonstrates bryostatin 1's promising therapeutic potential through the promotion of microglial signaling pathways that support oligodendrocyte maturation. And so figure B is clearly just the quantification, as I said earlier, of figure A. Now we can move on to figures D and E through H. And so I'd like to start off by saying that for figures E and H, these don't actually involve any techniques that I particularly was involved with, but they are important for understanding the story. So real remyelination within the spinal cord lesions of these LPC-treated mice were assessed at 15 days post-injury using immunofluorescence staining for MBP, which is a myelin marker, and SMI31, an axonal marker indicating healthy axons. And so increased co-localization of MBP with SMI31 positive axons was observed in the bryostatin 1 treated group, suggesting enhanced remyelination compared to vehicle-treated controls. So if we look at figure D, it's a bit hard to tell, but you can see that the BRIO1 group shows more co-localization of the MBP, which is purple, and the SMI31, which is green. And then the graph next to it is showing the statistical or the QPath result, the counted result for which group actually has more co-localization. And figures E through F are further confirmation using transmission electron microscopy, which revealed that bryostatin 1 treatment significantly increased the percentage of myelinated axons and improved overall myelin thickness. This is what is meant by the pooled G ratios. So these TEM findings also strongly support the therapeutic benefit of bryostatin 1 in promoting effective remyelination. The purpose of having E and H is to further corroborate the figure D results so that we can actually obtain some knowledge about the thickness or actual state of the axons rather than just having fluorescence to prove that. So this is just an interesting aside that I wanted to add, and it was a result of the figures and data that I presented before. But MS disproportionately affects females with roughly a 3-to-1 female to male ratio, and sex differences have also been documented in CNS immune responses and remyelination processes. Considering this, we analyzed bryostatin 1's effects by sex and found improved oligodendrocyte differentiation and remyelination in both sexes, although increases in oligodendrocyte lineage cells were specifically prominent in male mice, which is why I have a male mouse here to visualize that. But in all our in vivo studies, mice were randomized into treatment groups with separate investigators performing randomization, treatment, and blinded analysis to ensure unbiased results. In summary, my contributions to this project focused on evaluating how bryostatin 1, a PKC modulator, influences remyelination in a mouse model of multiple sclerosis. Using the LPC-induced focal demyelination model, we found that bryostatin 1 significantly increased the number of mature oligodendrocytes and promoted more effective remyelination. Through immunohistochemistry and transmission electron microscopy, we observed that bryostatin 1 not only improved oligodendrocyte differentiation, but also enhanced myelin repair, as evidenced by increased MBP expression and healthier G ratios. These findings support the idea that bryostatin 1 can shift microglia toward a regenerative state and promote tissue repair in the CNS. So, I'd like to close with future directions and a reflection. So, our findings identify PKC as a relevant target for future studies. This is because of the fact that bryostatin 1 is a PKC modifier. So that makes it an obvious source of more studies, and to be honest, our lab has started pursuing that as well as more bryostatin research. But these results also identify bryostatin 1 as a lead candidate for drug modulating microglia and CNS associated macrophages. Given the critical role of microglia and CNS associated macrophages in progressive MS and other neurodegenerative diseases lacking effective therapies, identifying treatments that manipulate these cells in a clinically favorable fashion remains a vital goal. And lastly, since bryostatin 1 has existing human clinical trial experience for other conditions, it's very promising for clinical translation, making all of this research in an animal model feel like it's still extremely valuable to human medicine and human health. Overall, reflecting on this research has helped me develop my wet lab skills, my analytical skills, and technical skills. It's deepened my interest in neuropathology and biological mechanisms, and it's given me great insight into the publication process and just being in a lab. I'd like to thank Dr. Payam Garabani and all of my lab members, Dr. Kornberg, my PI, for all of their help and for allowing me to use all of this data in my presentation. Thank you.